Good morning, everyone. Today we will explore the intricate relationship between the gut microbiota and obesity. I'm NUR binti Ramzi with metric number A197077, a clinical student in biomedical science, and over the next 10 minutes, we'll examine how a healthy gut community functions, what dysbiosis entails, the mechanism linking microbes to obesity, and how metagenomic sequencing is revolutionizing our understanding and treatment approaches. First, let's define a healthy gut microbiota. Approximately 60% of our gut is composed of diverse microbial species that perform essential functions. They ferment dietary fibers into short chain fatty acids, such as acetate, propionate, and butyrate, which serve as energy substrates for colonocytes and systemic metabolism. These microbes also synthesize vitamin K and vitamin B12, which is crucial for blood clotting and neurological functions, respectively. Furthermore, they reinforce the intestinal barrier and modulate immune responses, preventing pathogen overgrowth and systemic inflammation. In obesity, this balanced ecosystem becomes disrupted, which is a state known as dysbiosis. We observe an increased Firmicutes to bacteroidetes ratio. Firmicutes possess enhanced enzymatic machinery to extract additional calories from complex polysaccharides, thereby increasing energy harvest. Concurrently, dysbiosis compromises gut barrier integrity, leading to increased permeability. Lipopolysaccharide or LPS from gram-negative bacteria enter circulations, triggering chronic low-grade inflammation. This inflammatory milieu perturbs metabolic regulation in the liver and adipose tissue, perpetuating weight gain and insulin resistance. Let's dive into specific mechanism. Dysbiotic alterations impair short-chain fatty acid production and disrupt anorexigenic hormone signaling, where glutamyl levels rise while indole derivatives and bile acid declined. These changes interfere with central appetite control and the mesolimbic reward system, driving excessive calorie intake. Meanwhile, elevated LPS and reduced berry function precipitate endotoxemia, activating toll-like receptors and NFK-KB pathways that foster systemic inflammation and insulin resistance. Ultimately, this combination of hyperphagia and metabolic dysregulation underlies obesity development. Here, I've shown you the schematic illustrates multi-organ crosstalks of gut microbiota contribution to obesity development. Gut-derived SCFAs bind GPR41 and GPR43 on the enteroendocrine L cells, stimulating PYY and GLP-1 release, which normally curb appetite. This biosis attenuates these signals, so leptin and CCK regulation falters. In the liver, SCFAs modulate AMPK and FXR pathways, affecting lipid synthesis. In adipose tissue, LPS and SCFAs influence macrophage recruitment and adipokine secretion. Collectively, this shift tilt the balance towards fat storage, inflammation, and impaired insulin sensitivity. To dissect these microbial communities, we rely on shotgun metagenomic sequencing. We extract microbial DNA from stool samples and construct DNA libraries. High throughput sequences generate millions of short reads. Bioinformatic pipelines assemble these into contigs, reconstruct whole genomes. This approach provides species and strain level resolution and a comprehensive catalog of functional genes. We can map metabolic pathways, identify novel contributors, and pinpoint targets for precision interventions. Applying these technologies to obese versus lean cohorts reveal distinct microbial profiles. Certain taxonomy, like increased firmicutes, correlate with energy extraction pathways, while others associate with pro-inflammatory gene clusters. Functional annotation highlights genes involved in SCFA biosynthesis, bile acid metabolism, and endotoxin production. These microbial signatures not only stratify obesity risks, but also serve as biomarkers to monitor interventions and predict outcomes. In summary, gut microbiota dysbiosis plays a pivotal role in obesity pathogenesis by altering energy harvest, hormone signaling, and inflammatory pathways. Metagenomic sequencing has illuminated specific microbial and genetic targets, paving the way for microbiome-based precision therapies, ranging from tailored probiotics to dietary modifications. As clinical students and researchers, integrating microbiome profiling into obesity management holds promise for more effective, personalized interventions. We have moved on to the last slide. This is the key references for today's presentation. Thank you for your attention. Goodbye.